Okay, today for the Journal Club, we're going to discuss a paper by Jules Freund, that's spelled Freund, and Katherine McDermott from 1942, which is, of course, one of the World War years. It's published in the Proceedings of the Society for Experimental Biology and Medicine, which was one of the major biologically, medically oriented journals at the time. Now we have a plethora of those kinds of journals, but back then there weren't very many. And the title is The Sensitization to Horse Serum by Means of Adjuvants. And so what they were studying was trying to figure out how to really markedly potentiate the immune response using horse serum as their antigen. And of course, just to give you a little bit of background on that whole thing, remember now that 1893 was when William Coley was trying to cure cancer with injections of bacteria, killed bacteria, but live bacteria and killed bacteria. And then, of course, I mentioned last week, in 1924, two investigators from the Institut Pasteur in Paris reported that they had generated a vaccine for tuberculosis called BCG, for Bacillus calmette-guerin, which was ostensibly a bovine tuberculosis organism. And so TB obviously was a very big deal back in the 19th century, and also for the whole first half of the 20th century. When I went to medical school in the 1960s, they had a whole separate building for tuberculosis. That just shows to go you how much of a problem it was. Now then, the background on the authors of this paper, Jules Freund, was a typical story that we've all heard in the United States about the American medicine and science capitalizing on the wars of the 20th century. And of course, World War I from 1914 to 1918, the Austro-Hungarian Empire became no longer. And it just turns out that Jules Freund was Hungarian and had graduated from medical school in 1913. So he born in 1890. And of course, 1890 is the year that von Behring and Kitasato reported the first evidence of antibody activity in serum. And then as we went on, people started using hyperimmunized sera for therapy, for diphtheria and tetanus and so forth. And so the antigens that people used in the first half of the 20th century were basically not purified proteins. And of course, we didn't know what proteins really were at that particular point in time, chemically anyway. And so they just, they used whatever they had around. So they had horse serum. So that became the antigen. The other thing they used was hen egg white, or ovalbumin, the albumin in the egg white. And so egg whites were mostly albumin, but there were also a lot of other proteins involved too. So it wasn't a homogeneous, purified single molecule. It was a whole gamish of things. Freund graduated from medical school in 1913. And of course, the war started in 1914. And so he was in the army during the war. And the good news is he didn't get killed. And so he mustered out. For the first 4 or 5 years, he worked at the university as an assistant professor, and the same university, the Royal Hungarian University in Budapest, upon which he had graduated. But Austria couldn't have been a very good place to be at that particular point in time. And so he found his way to the United States of America. And that's why I say it's one of these sort of typical stories that we've been able to benefit from all these immigrants. And of course, in the last several years here in the United States, we've been questioning whether or not we will be winning these immigrants, particularly the non-white variety. But Freund being of the white variety, he came in. He got a fellowship at Harvard, and then he went on there for a couple of other postdocs and so forth. Ultimately, ending up in Philadelphia for 5 or 6 years at a research institute studying tuberculosis. And then in 1932, he found his way to the New York City and to the Cornell University Medical Center, which was, of course, my medical school and medical center in the last 25 years of my career. It was from there that he wrote this paper that was published in 1942, because he was there at Cornell until 1945. And then he went on, he had an illustrious career in immunology. He ended up being recruited ultimately to the NIH in 1957. And of course, that's the year that Sputnik was blasted into space by the Russians, and which then essentially opened the floodgates of funding for biomedical research in our country. But the bad news is that he didn't last very long, even though he was the first chief of the immunology lab, the Laboratory of Immunology at the NIAID, from whence Tony Fauci was also who is now the director of the NIAID, just to give you some sort of a context of everything. Because he died in 1960 of multiple myeloma. So isn't that the irony of the whole thing? Multiple myeloma, of course, myelomal cells, plasma cells, when they become malignant are called myeloma cells, myo meaning bone. And so they come from the bone marrow, and they secrete immunoglobulin and so forth, for example. But he died then in 1960. So between 1890 and 1960 is when we're talking about this. And so in the introduction of this paper, Freund and McDermott go through what was going on in the business at the time. And of course, in 1924, just the same year that Calmette and Guerin published their paper on a TB vaccine, it was reported by investigators that if you took guinea pigs that were infected with tuberculosis and immunized or immunized them with a particular antigen, and then had a control group of guinea pigs that did not have tuberculosis, that the ones that were infected with TB made much more antibody. That was good. And so that's what really set them on the road. So that's 1924, 1928. You know, they did experiments. People did experiments to see if killed TB would work just as well as live TB, because you didn't want to be fiddling around in the laboratory with all these bugs, live bugs all the time, and particularly because you can breathe it in through your lungs, and that's why it was contagious essentially. And then in 1937, Freund, along with other investigators, was trying to figure out ways to boost immunoreactivity. So he used these data from the 20s that tuberculosis somehow was immunoenhancing. And so he did experiments with paraffin oil and killed TB, that he suspended the TB organisms in this paraffin oil, which was a petroleum oil essentially. And he found that if he inoculated guinea pigs with killed TB organisms in paraffin oil, that he could get an immunological reaction as measured by a skin test that nowadays we still use this skin test. It's called a PPD test, for purified protein derivative. And you put a little bit of that just into the skin, intradermal inoculation, and then 24, 48, 72 hours later, you would get a little circle of inflammation at the site if you had been infected with tuberculosis. If you hadn't been infected, even though you'd been exposed, if you hadn't been infected, meaning that the bugs didn't get inside of your body, you wouldn't have an immune reaction. And so this became the sine qua non for so-called cell-mediated immunity eventually. But Freund was using it just as a test to see whether or not he could get guinea pigs sensitized to TB and other antigens. And he tried it, they tried it with ovalbumin, egg white. Into using the paraffin oil and the TB organisms, mixing it then in with hen egg white, and then testing later on with hen egg white and also TB organisms. He could get PPD, he could get a skin reaction to PPD, but he couldn't get one to the ovalbumin. And so that set the stage for the particular experiments in this paper. And so that this paper really is the paper that everybody refers to nowadays for what is now known as complete Freund's adjuvant. And I'll get into that, what that consists of. So Freund, he figured that he also looked at some other stuff was going on at the same time in the same place in New York City at the Rockefeller Institute. Carl Landsteiner, who was a very famous one in immunology and in serology and so forth. And another fellow, a younger fellow by the name of Merrill Chase. They were studying contact sensitivity, and that sort of like poison ivy. All you have to do is have contact on your skin with some kinds of chemicals and so forth. And the favorite became picryl chloride. And they were trying to sensitize animals to picryl chloride. And they found that if they put the picryl chloride in with paraffin oil and killed TB organisms, but also other antigens as well, in addition to picryl chloride, they could get skin reaction, a skin test positivity, a cellular reaction to that whole thing. So Freund and McDermott said their hypothesis at this point in time is that if they thought that the problem with protein antigens like ovalbumin, and the reason they couldn't get a reaction as easily as they could with, say, to the TB organism and so forth, is that when they put it into the paraffin oil kind of thing, maybe the protein got separated somehow from the oil and so forth. So that what they did was they thought that maybe if they would mix the antigen in saline, water soluble situation, and then add an emulsifier. And for an emulsifier, they used lanolin, and lanolin, I had to look it up, but lanolin essentially is esterified fatty acids, and it acts as it's like soap, acts as an emulsifier, so that when they've got the water soluble thing, they got the emulsifier, and they thought that if now we put this with paraffin oil and TB, this is going to work. And so that's what these experiments are about. So they chose horse serum as their antigen, and they mixed it together. They combined it then 10 milliliters with the emulsifier, the lanolin, also 10 milliliters, and they put this in a mortar and pestle, and they ground it all up. And then they added 20 milliliters of paraffin oil that it already had suspended in it that killed TB organisms, and they mixed it up some more in the mortar and pestle. And then they used a dose of a 0.5 ml of that solution for each immunization. They immunized guinea pigs intramuscularly at the back of the neck and the paraspinal muscles at the back of the neck. And then they waited various periods of time thereafter, and they then would shave the back of the guinea pig or the stomach of the guinea pig and inject small amounts of horse serum in saline into the intradermally to see whether or not they could get a skin test positive reaction. And initially they did 1, 2, and 3 weeks after the sensitization in the back. And then they waited a year, and they did it for a year longer. And as a control, they used just horse serum in normal saline as for the sensitization phase of the whole experiment. So that's what they did. And all the data are in tables. There's 3 tables. And I didn't try to put them on screen sharing because my reproduction wasn't the greatest. And I figured that, and the data are simple enough, you can just glance at the tables and figure out what happened. In table 1, when they did the skin test reaction using horse serum and normal saline, they got positive reaction in the animals that had the antigen horse serum given together with what they called adjuvants. Now, what an adjuvant, the definition of an adjuvant is anything that potentiates the immune response. But they didn't get any reaction in the control animals immunized just with protein and normal saline. Then in table 2, they did this for various time intervals, 1, 2, and 3 months after the sensitization. And that was in table 1. And then in table 2, they had the results after a whole year. They waited a year, and then they tested this guinea pig for the skin test reactivity. And again, they only found positive reactions in the animals that got the whole gamish. And so finally, in table 3, they after 1 month, they bled the animals and they looked for antibody activity in the sera of the guinea pigs. And what they looked for was a precipitin reaction, so that under the microscope, if they mixed a little bit of horse serum with the antisera with the serum, they would see flocculation or aggregation under the microscope. So that was the precipitin reaction. And again, they only had positive results in those animals that got the adjuvant, the antigen in the adjuvant. So they go on in the discussion in this paper. And the first thing they say is, well, the mechanism of action of adjuvants is complicated. And of course, this is where I started my career back in the day. And I remember one of the parts of my story that's actually in my new book that I'm serializing in my newsletter, in case you're interested. Chapter 2 deals with my days with Georges Maté in Paris. And when I first discussed what my projects could be, I told him I wanted to understand the mechanism of action of adjuvants. since they seem to be so important for the anti-cancer effect that he had reported. And then he just laughed at me and said, it was too complicated for such a young buck like you, Smith. You can't do that. You have to, you need to know more stuff. So I thought it made me smile when I saw the first sentence of their discussion. The second thing they said was that it was clear from their experiments that killed TB and live TB were equivalent. You didn't need to have live TB, which was a good thing. Question is then, what was it? And of course, TB was discovered by Robert Koch back in 1894 because he had devised very, very unique experiments to stain tissue samples from people infected with tuberculosis. The tuberculosis bug is a very tiny little guy. It's about 1/10 the size of the anthrax bacillus that was also used at the same time back in the 1890s. But the coat of the mycobacteria, which is the genus from this species is taken from, is a very waxy, lipid-containing, difficult thing to work with and difficult thing to stain under the microscope, which is how Koch finally figured out a recipe that he could do that. So something had to be with in terms of the adjuvant effect of these TB organisms had to have been wrapped up in that whole process. They said on the basis of the experiments done by others and also their own experiments that the paraffin oil, which seems like a funny thing to stick into people or guinea pigs, but that increased the cellular reaction, the skin test reactivity, the cutaneous reactivity. And they just speculated that perhaps the reason was that the paraffin oil, the oil coating of these bacteria somehow prevented them from destruction and so forth, and it sustained then the antigenicity over time after it was injected in the sensitization step of the whole deal. And then the emulsifier, the lanolin from the sheep's wool, they figured it must have retarded the separation of the horse serum, which was water soluble, from the lipid paraffin oil kind of a situation, and kept everything together longer so that they could get an immune reaction. But they didn't really know what was going on, what was happening. But these are the beginning experiments. These are the kinds of experiments that were done in the first half of the 20th century, when investigators were simply trying to figure out what is an immune response. And so as a consequence of all this, so that was their interpretation of their experiments. Their conclusions, they also have in this paper, were a little bit redundant. Killed TB is equal to live TB. The duration of sensitization when you use this adjuvant business is a long time, up to a year, which is a good thing. The adjuvants are active in increasing both the cell-mediated immunity, the cutaneous skin test reactivity, as well as the antibody, the humoral immunity, the antibody immunity, which was also a very good thing. And they concluded in the end that the use of the emulsifier, the lanolin from the sheep's wool, was very important when you're using protein antigens and you wanted to get an immune response. So from there on after, for the rest of, up until the present time, if you're working in the laboratory and you're trying to get an immune reaction in animals, and of course, now everybody uses mice. You use complete Freund's adjuvant, which is this whole gamish of the water-soluble antigen with the emulsifier with the paraffin oil with killed TB organisms in it. So we still use that, get a great immune reactivity. You also get necrosis of the tissues at the site of the injection. And so it's not innocuous. And we don't use it in humans for that very reason, because it's too toxic. It was found then subsequently that after you immunize the first immunization dose with complete Freund's adjuvant, if you came back then and you left the TB out of the whole gamish, the TB bugs, the killed TB, that's called incomplete Freund's adjuvant. Still has the water-soluble antigen, still has the emulsifier, and still has the paraffin oil. And so we used complete Freund's adjuvant when we first immunized mice with interleukin-2 after we painstakingly purified it over months and months and months. And we then emulsified that in with the paraffin oil and so forth and the TB organisms and stuck it into mice and hoped and prayed after that. That was when I was testing God again to see if he'd answer my prayers. He did this time. He never did it when I tried to get him to make the Little League baseball team, though, you know. So anyway, that's the end of that. That is a very, very important paper in immunology. And of course, that led to, in modern times, the question was, why did you need all this stuff in order to get an immune response? Why can't you just put the antigen into the animal or the human and so forth? And of course, we're using one of the vaccines for coronavirus right now is it employs an adjuvant to boost up the immune reactivity. I can't remember which one, but there's maybe one or two out of a half a dozen or more. I think there's about 40 vaccines that are under investigation right now. So I think that does it. And I'll stop there. And don't forget to check out the newsletter. And don't forget to like this video and come to the Journal Club once a week, usually on Mondays on YouTube. Thanks. So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.